Welcome everyone to this third of four field days that we have. These are web seminars that will get everyone, one, excited about a upcoming workshop that we're hosting in Orlando that's adjacent to the AGBT Ag meeting to discuss how we can use single cell applications, how we can build our own community and apply these really exciting technologies towards agricultural applications, including plants and animals. The workshop, which I believe registration has closed for now, is going to host a range of really exciting speakers and also include a lot of opportunities for discussion, including a bunch of different breakout sessions to discuss how we can best use data, share data, create analytical tools, and also identify where the needs of the community are in terms of analysis pipelines and benchmarking. So would be very excited to see all of you there in Orlando. As I mentioned, this is one of these field days where we're discussing some of these topics ahead of the larger conference to get a better sense of where the community is at. We've had two really successful ones already in February. Today, we're going to be discussing different strategies for assigning cell identity in single cell data sets. And to help us out, we have two really talented and exciting speakers. The first is Song Lee from Virginia Polytech. And Dr. Lee is an associate professor at the School of Plant and Environmental Sciences at Virginia Tech. His group works on the application of machine learning and AI in plant genomics and phenomics. In the past few years, his lab has published machine learning methods for identification of gene regulatory network and novel marker genes using single cell data. In this talk, he will discuss a newly published method called orthomarker gene groups, or OMGs, to quickly assign cell types across diverse plant species without the need of data integration. So before I invite Sung to share his talk, I also want to shout out that next week we will also host our last of these field sessions that will focus on single cell data for advancing genome to phenome. And so I hope to see you all there as well. So Song, can you take it away here?
Yes, thank you for the nice introduction. I'm going to try to share my slides and going to do everything. You're seeing my slides, right? I'm going to get my laser pointer here. You're not seeing the presenter view.
Yes, we can see your the right screen. Yeah.
Okay, thank you very much. Thank you for the organizer for inviting me to talk about our recently published approach for mapping single cell identities across different plant species. You can go to the website here, and you can actually go to the GitHub repository in one of the link from our group repository to get it's moving. So, okay, to acknowledge people who did this work first, the two major contributors are Chuan Chao or Nina, a third-year graduate student in the bioinformatics program at Virginia Tech. And Sai now is working at Bloomberg as a web developer, but he developed the OMG website, which is our Shiny website, and funding source from NSF, Eager, and PGRP program. And other people contribute to this work, including the past postdoc, Prakash or PR, and Sanchari, and the co-PI from this grant, Bas Bergman. Again, you can actually, or you cannot click, but you can go to this link to see the code and the website we are using for doing this analysis. And so this is the presentation outline, and I'm going to spend most of the time talking about OMG work. And if I have time, I'm going to talk something else. But I did this practice several times, and I think I don't have time to talk about this. So let's get started. And why are we doing this single cell cross species matching in diverse plant species? And how did we do that? And this is moving again. So the goal for plant research, I believe, in the past 5 years and the coming years would be how do we leverage this new technology of single cell sequencing and to benefit the broader plant community, which means we want to be able to first annotate cell types across many different plant species besides the model species, And if you look into the literature, how people did this, the default method would be to do data integration, which was mostly used in human, mouse, or biomedical research. And there's many other type of approaches people use in the publications. So I'm showing you two different UMaps. One is a good example of integration-based assignment. The other one is not so good, which is what we tried and didn't work well for our case. And let me show you this one. It's just giving me trouble to switch slide right now. Okay, so this was published in Nature by Ken Berman's group a few years back. Basically, single-cell sequencing of 3 grass species: maize, Soteria, and sorghum. And on the left, you see the single-cell map, each of the cluster annotated by different cell type identity. On the right, you see the integrated single-cell map from 3 species. What you're looking for here in general is that you want to see the different dots represent different cells and different colors of different species. You want to see the dots from different species sort of mixed well in each of these separate clusters from all 3 species. In this case, you do see that, but except for there's a little bit outlier from maize in the middle. And the reason this integration works and it can be used to assign cell type is because the same group published a different paper a few years back using maize as their model system to generate single cell data, where they use a number of different techniques to label the cell types in the maize single cell map. And then once you have that one species single cell map generated, and then you have a very closely related species, you do integration, you can basically do sort of annotation transfer based on the overlapping clusters between species. We also tried similar approach for a more diverse case. And like in this case, we show an unsuccessful integration of different species. Why do I call that? We take the published Arabidopsis, rice, and maize single cell data, and then do an integration using Thread default pipeline. And what you can see, you still do see that the 3 species dots are overlapping with each other, mixed pretty well in the single cell map. And then we start to find all these clusters. These are just typical integration-based pipeline from Thread. And the next thing is that we look at the actual cell type identity based on this integrated map for each of the cluster. And then this is what I see. And what happened is that we took all the annotation for each of the cluster identity based on the original publication, and then put them into these integrated clusters. Out of 19 clusters, only 2 of them are homogeneous clusters, which means they are from the same cell type from 2 different or 3 different species. All other clusters in this integration coming from different cell types, different species, there's just a mix of different cell type. That's why we think this is not very successful integration. And so with that, we move on to using other approach for doing across species single cell type annotation. And again, in the literature, we summarize the way people do this. There are basically 4 different ways of how do you assign cell types in different plant species. And we summarize that in the book chapter we published earlier this year. And we are particularly interested in marker-based approach. The reason for that is because some other approaches are not generalizable for cross-species comparison. And this was a collaboration with Sean Brady's group a couple of years back when they generated single-cell sequencing for tomato root. And the reason we can annotate this is because the same group published, again, marker gene-based single-cell sorting, but bulk RNA-seq. Well, let me take that back. So they have the marker lines for different cell types or cell layers in tomato root already generated in the previous publication, and they sorted those cells and generated bulk RNA-seq data from those sorted cells. And then we get a long list of cell type specific markers for different cell lineages in tomato root. And that is when the postdoc Prakash or PR did this analysis, taking those marker genes from their cell paper and then overlap with the marker genes from this single cell map in tomato by Alex. And then we did a statistical test to find the significant overlap between those markers. And then we were able to assign some of the clusters. And Alex actually took this a little bit further to use some other approaches to assign cell clusters and then put them together to annotate this map of tomato single cell data published last year. And go a little bit way back in the early stage when the single cell data was first published in plants in Arabidopsis and rice. A grad student, Haidong, he was a grad student and postdoc in Bob Schmidt's lab. Now he's a faculty in China, one of the Chinese university. And he looked at the single cell markers across Arabidopsis and rice in the first batch of published single cell maps. What we find out is that if you look at this row, the known markers in cortex have 300 plus in Arabidopsis, 600, close to 700 in rice, but there's only 10 overlap between these two. Some people are okay if there's some really significant cortex markers that sort of biologically conserved. People are okay with this 10, but then if you do a statistical test, the p-value is not significant. That is when Haidong had this idea of using machine learning to just increase the number of markers can be identified in Arabidopsis and in other species, and he basically increased marker to about 2,000 of them. We end up with 93 overlapping, and the p-value becomes significant, significantly enriched. So the good news is that it's significant, but the downside of this is that it's 90 plus genes out of over 2,000 markers. So the take-home message is that less than 5% of Arabidopsis one-to-one orthodox markers in rice are conserved. So you don't have, again, 93 divided by about 2,000. So that's where this 5% come from. Also, the challenge is that if you use 2,000 markers, many of them are essentially not that specific to a certain cell type. We don't want to use so many markers from one cell type. So this is just one example from cortex. What do you see if you look at all other clusters, right? And this is the outcome of that. We take the one-to-one ortholog between rice and Arabidopsis based on phytosome, and then we take the overlapping markers between the clusters, and these numbers indicating how many one-to-one orthologs are overlapping out of 200 markers. And you can see the xylem, non-hair cells are matched, and then phloem. That's 3 out of 8 significant overlaps are from sort of orthologous cell types. And there's many other overlaps between different species, different cell types as well, but they are not statistically significant. So that is when Nina came on board in 2022-ish, and where we decided to solve this problem using this also marker groups pipeline. So the pipeline is simple. To begin with, follow the typical thread pipeline where you generate these clusters. You can use other type of tools as well. Generate these clusters for your species. You have a reference species and query species. And then you call these marker genes, again, follow standard thread pipeline. And then typically what we do is we first using this program called also finder to generate also groups. And then we find the one-to-one ortholog genes. And then we do this statistical test. But the insight from Nina's work, or after many failed attempts to try to identify the real best one-to-one ortholog genes, is actually just get rid of this step. We are not counting one-to-one ortholog genes. We are counting this also group. To put it another way, if you have 2 genes, the same gene in Arabidopsis, and you have 1 ortholog in rice, to those 2 genes, you count all of them in one single also group. And then you do that also marker test using statistical enrichment. And we use 200 markers per cluster. I have a slide to show why that is the case. But we don't want to use too many markers, because if you have too many markers, the enrichment, the fold change of those markers become very, very low. Okay, so what do you see? So go back. This is the one-to-one ortholog genes case. If you do this test between 2 species, rice, Arabidopsis, 3 out of 8 significant overlaps, and there's some errors in here as well. And if you do that OMG method, also marker groups, you suddenly increase the number of overlaps that are correct. So again, one-to-one orthologs, OMG, and that's why I call it OMG, right? You have hair match with hair, cortex match with cortex, xylem match with xylem. Instead, there's many other overlaps of markers, but only the ones that are exactly ortholog tissues are or cell types are significant. And there's one case where cortex-like is annotated in rice, which is annotated endocortex in Arabidopsis. So we count that as another good match between 2 species. Okay, so by this time, I think most of you are probably looking at these 2, which I highlight here. These are really high number of also marker genes overlapping between non-hair cells and still in Arabidopsis, and meristem and endodermis in rice. So what are these? Are these mistakes? Yeah. So it said maybe they are wrongly labeled cell types. Actually, we pull out these genes, 200 marker group, 200 markers, and did the enrichment test using Go enrichment analysis. What you can see very interesting. This is Arabidopsis clusters. Look at the non-hair one, non-hair two, non-hair one, non-hair two. Their Go term are very different. Interestingly, the non-hair two has exactly Go term enriched as the still one, which are basically these two arrow indicated. But more importantly, if you look into the enrichment of Go terms for the rice genes in the endodermis and the meristematic zone, they are the same Go term enriched in these two as compared to the non-hair two and still one, which are basically ribosome genes. So sort of an indicator of some sort of ribosome quickly replicating or meristematic tissue are the actual identity of these cell types. So the hypothesis would be that these clusters are basically wrongly labeled by the original publications. They should have been labeled as meristems instead of the other identities. I have talked about this multiple times. I talk with other people, and actually different people have different hypotheses. For example, some people think these are low quality cells, and some people think these are a protoplast artifact. I don't have a slide to show this, but if you look at the actual where these clusters are in the UMAP, they're actually in the center. It's like a big cluster 0 or 1, which typically are indicator of sort of a meristematic or some sort of undifferentiated cell clusters. So, and another potential hypothesis is that they didn't, the original author didn't label them wrong, but these are basically meristematic non-hair cells. For example, they have the non-hair cell markers expressed, but they also have a lot of meristematic markers expressed. So it's sort of dual function or the same type cell type with different developmental stage. So those are the hypotheses we have about this observation. And to quickly summarize 15 minutes, the most important thing is that we need to do a statistical test to assign your cell type based on the marker genes. And I showed you this orange arrows are the cases where there are potentially mislabeled cell types from publications or their dual function cell types. And I'm going to show you what these open arrows are in the next few slides. But just to quickly summarize, our current method enable cross-species between monocots and dicots comparison in plants. This is a marker-based method, so it's very fast. You don't need to do very computational heavy integration. We use thread output as default input for the method. And we have a website, you can upload it, or you can run the R package if you're comfortable with those R command line. So statistical test, why that's important. I'm showing this figure because many people also show this in the publication. This is one of our own paper. So what people usually do, this is called dot plot, right? So each of the column is a gene and each of the row is a different cell type. And then people will say, okay, we find these 5 marker genes really highly enriched in this cell type. So this is a really good candidate for this marker. Typically, people will point out 1 or 2 genes are well-known markers for certain cell type, and that's it. That's how the cell type were determined, couple years back. In many cases, including our own paper. This figure shows that that's probably a not so great way of doing it. Using xylem as one example, we have 43 marker, these OMG groups overlapping between this xylem in rice and xylem in Arabidopsis. But if you only show 5, right, and even 10, you can assign still phloem or hair cells as xylem cells, right? So that's what I'm trying to explain, that if you pick 10 genes, these 10 genes are markers including a known hair cell marker, then you probably could potentially mislabel xylem as hair if you ignore this 43 overlapping in xylem. Same thing happens for the rice. The rice xylem cells could be wrongly labeled as cortex or hairs or some other cell types. So that is where it's really important to just not only show this, but also show this across all your clusters when you use markers to assign cell types. And so that's the reason for using statistical tests. The next one is something interesting, that when you compare different plant species, there are cases where you start to see cell types that are not in the other species, which in this case is the exodermis, right? So the exodermis is a cell type that exists in more than 80% of land plants, but they are just not there in Arabidopsis, but they are in tomato. When we have tomato single cell data compared with Arabidopsis, look at exodermis, it actually maps to these two clusters, which are endocortex and endodermis for Arabidopsis. And this other exodermis cluster maps to a cortex cluster in Arabidopsis. The good news is that these are all considered as ground tissues. So they're not mapping to xylem or phloem, which are vascular tissues, and they're not mapping to hair, non-hair, which are epidermal tissues. But the downside is that if you have an exodermic cluster in tomato, but you find them overlap with endodermis or cortex, you might wrongly label them as endodermis or cortex. The reason this was labeled as exodermis was basically because the cell lines was available for tomato analysis. That's one reason we have this already known markers for exodermis in tomato. The second is that if you look at the UMAP, these two clusters are actually together, and they're away from the two other clusters. One of them is clearly cortex, the other one is endodermis. So that is another case where when you have cell types that are across different species that are not represented in your reference map, you may have some mislabels. So how do we deal with this type of issue? And the power comes from so-called meta-analysis, where luckily by 2023, when we did this last batch of analysis, there was a Chinese group published a database summarized 15 different species plants single cell data, and they generate markers and provided tissue labels for those markers. And then we put them together using the OMG approach. We end up with this huge map of close to 1 million cells, 268 clusters from 15 species. And then we find these subclusters within this map. For example, this big cluster here with this red blob are all meristematic tissues or cells, and there are about 10 times more meristematic cells enriched in this cluster. And you have smaller clusters, 1,500 times more enrichment in the G2M phase, and you have the S phase enriched 260 times. So we have these subclusters. Not all the cells in this cluster are meristem, but then if you do a statistical test again, you'll see a significant enrichment. Out of 15 clusters we get, only one has a mixed identity. Others, we can assign this identity very clearly based on enriched cell type, like root hair, phloem, and inflorescence tissues. So again, you can go to the GitHub page to see how we did this, and the code are all there, and the link to the website is also over there. With that, how do we use this? Focus on the top portion. And if you have a reference map, we already integrate into this 15 species map, and you have a query cell map, you don't know what zero cluster is, you just do the statistical test, and it will put on the map and tell you what are the most likely cluster based on the marker overlapping. And this is how you do it. This is the OMG website. First step, pick your species. The key step here is that you need to know the gene pattern is the same as what we use, or gene ID is the same format as we use in the website. And then you put in your CSV file, which are the marker files I'll show you in the next slide. This is basically a thread output. And then we are going to do, you're going to pick the reference species, and then this will give you the enrichment markers here. And the next one, this is what this looks like. The CSV file is basically thread marker files. And then once you click on this overlapping marker, in this case, cluster 5 is a meristematic zone. The website will give you what are the markers from tomato, and you sort it the other way, you get the Arabidopsis markers, and they give you the log fold change, and you can go ahead and look at their biological functions. And I'll give you one more example we have been doing in our group, is to use this for pathogen infection identification and to see what are the pathogen infected cells. We did a root infected with certain plant pathogen, which you will see in probably a year what we're doing. And then we have a cluster that is not annotated by OMG to be aligned to any clusters, which is cluster number 5. And what you see here, you compare Arabidopsis root with other species root. Cluster number 1 is a clearly meristematic zone cluster here. And you compare that to tomato, you still get meristematic zone enriched here. but you don't see anything enriched for cluster number 5 at all. Next, you look at rice, you still get the meristematic zone enrichment here, but you start to see overlapping with this cluster 1 with endodermis in rice. And when you move to maize, the meristematic cluster is gone, but the endodermis remains. That means this cluster number 1, which is not the pathogen infected cluster, may have some endodermis marker genes in there in Arabidopsis. However, what you see is that you don't see any cluster overlap with cluster 5 in the root data from any plant species we have in the database. And once you put that beyond plant to overlap with shoot apex and leaf, this is where the power of this overlapping OMG method start to show, where when you compare Arabidopsis shoot apex with root, you start to see cluster 5 have this meristematic or proliferating cells identity. And then it also have the overlapping markers with ribosome identity with tomato, and proliferating cells from populus, and also meristematic cell from this flower species. So that means this cluster 5 is not as similar to any of the root clusters, but they have some meristematic activities going on, which could be the effect of pathogen infection. So to summarize that, this is what we are doing using OMG. Basically, originally we think this is to be used for non-Arabidopsis species mostly, but we can find use of that for analyze Arabidopsis data set as well by extending this comparison to many other species. We are working on things that are enable people to use this across species that are not in this 15 species. We are trying to do a blast OMG version, but the student really want to do a large language model version. So we are working on this too. And what else do we want to do? We want to do connect this with breeding community so that we can do genetic engineering or GWAS to use this information we gained by cross-species comparison to help people in other research domain in plant science to get better understanding of their gene functions. And finally, we don't have the personnel to actually do this in non-plant systems, but I'm curious to see whether this works in non-plant system as well. With that, the limitation would be how do you decide number of clusters, markers, and different species name the cluster differently. And for us, it's very hard to reproduce, in many cases, the same published UMAP clusters, basically because the detailed parameters were not provided in the publication, which is understandable to some extent. And these figures show why we pick 200 markers. That is when the number of overlapping OMG start to take off, well, the fold change of markers are still above 1, log 2 fold change, which means twofold. And if you go more, the log 2 fold change of the markers become much smaller, although you can have more overlaps over different species. With that, that's all I want to say. Importantly, you want to do a statistical test across all the clusters in your single cell map before you assign the cell type using some type of statistical test. OMG is one of them. And the last slide would be that we are actually open a summer camp for helping you to incorporate your data, single cell data, into OMG framework. If you're interested, send me an email. I'll put you on the email list for this summer camp. This is funded by NSF PGRP program. With that, I can take any questions from the audience. And on my count is 25 minutes.
Awesome. Thank you, Song, for staying under time. I think we're actually going to hold questions until our next speaker. So I also want to introduce co-host Fiona McCarthy, who will introduce our next speaker.
Okay, thank you, everyone. Thank you, Ben. It's my great pleasure to introduce our next speaker. Dr. Chuan Zhu received his PhD from the Joint Chinese Academy of Sciences Max Planck Partner Institute for Computational Biology. And he did his postdoc at the Yale School of Medicine in 2018, I think that was. He joined the Teichmann Lab at the Cambridge Stem Cell Institute in 2020. And this is where he's been studying the principles behind complex tissue organization. And of course, with that comes his single cell work. Most His work includes developing cell typist and cell hint software to identify cell types in single cell data. And this is what he's going to be talking about today. So Chuan, if you would share your screen. Thank you very much.
Yeah.
Great, that looks perfect. Thank you.
Yeah. Yeah, thank you for introducing me. It's my great pleasure to present at this meeting. My name is Chuan Xu, a computational postdoc at the University of Cambridge. And today I will share with you our work on automated annotation and integration of single-cell data using machine learning tools, which we call cell typist and cell hint. Let's think a fundamental question in tissue biology. What's the first step towards understanding human organs? The answer is probably to understand its genetic makeup. Two layers of information are necessary here. The first is the genome sequence, which was made available through many years of efforts, such as the Human Genome Project, providing the molecular basis for downstream biological explorations. Accordingly, single-cell genomics provides the cellular basis for understanding the cellular composition of the human bodies. One thing I would like to note is that disentangling the cellular composition of humans is not only about finding the full list of cell types in the body, but also about its anatomy, developmental history, functionality, and cell type interactions, which as a whole constitute the cellular landscape we have seen. And today I will focus on two basic questions. The first one is how to annotate cell types on the basis of single cell data. And then after cell types are annotated in a single dataset, how can we unify different cell types across different datasets? To answer these two questions, a natural answer would be we need knowledge to annotate cell types and knowledge to unify cell types. However, usually we do not have enough knowledge. And even we have, this process is very time consuming. Therefore, we need data to complement our knowledge. Also, from the perspective of machine learning, big data is crucial. For most single-cell tasks, there is a trade-off between cell type knowledge and the amount of data. Specifically, when you have limited data, you have to rely much on the knowledge. On the other hand, when you have more data, the demand for the knowledge will be correspondingly reduced. Building on this idea, we developed cell typist to annotate cell types based on single cell data, along with a cell type database built on cell typist. So what is cell typist? Cell typist has a very straightforward workflow. First, a reference data is trained into a cell typist model, and then this model is used to classify query data into different cell types. Importantly, among different algorithms, we built cell typist on logistic regression, a canonical machine learning algorithm, because it has a linear decision boundary at a high dimensional space, as well as other advantages I will touch upon in the following slides. Actually, you can imagine that with only a handful of genes, cell annotation and classification is a relatively simple task. So with cell typist, our ultimate purpose is to resolve the high dimensionality imposed by tens of thousands of genes in the single cell data. Our first focus is interpretability. Using logistic regression, cell typist build a linear hyperplane to serve as the decision boundary, which can be used to classify query cells into different cell types. This process naturally involves an probability output, which can be used to enhance the interpretability of our algorithm. And next, to increase the robustness of this decision boundary, as mentioned earlier, we need to have more data sets to increase the feature space coverage and to stabilize the decision boundary. I will use immune cells, which was the initial focus of cell typist as an example. And we collected 19 immune datasets from different sources as listed here. And some of these datasets did not contain pure immune cells, so we extracted immune cells from these datasets. Through modeling these datasets using cell typist, you can see a clear trend that when we collect more cells for a given cell type, its cell type prediction accuracy will also be increased. So in total, big data naturally empower knowledge acquisition. In addition to interpretability, a good model is supposed to be lightweight and shareable. And cell typist has such property because using logistical regulation for each cell type, instead of recording a long expression vector, we only recorded the gene weights, thereby reducing the size of the model. A byproduct of this process is that we can leverage the gene weights to derive the so-called cell typist cell type encyclopedia. Within this encyclopedia, we enumerate all cell types, and for each cell type, we have detailed information such as cell type description, T-cell origin, as well as marker genes derived from the gene weights mentioned above, meaning that you can retrieve different kinds of information all relating to a single cell type. To further reduce the size of the model, cell typists implement a feature selection process to automatically select genes with high weights to further reduce the size of the model. Here shows some example cell typist models, and you can notice that compared to the original data, which can be hundreds of gigabytes big, the model sizes are significantly reduced, meaning that you can easily share models with different users. And the last thing to consider is that the reference data and the query data usually do not have the same set of genes. And what cell typist does is to use the intersected genes to result in a reduced hyperplane, namely classifying cells within the overlapped space. This is actually a very simple approach, but has great application potential because now cell typist models can be repeatedly used. Building on this, we developed an online interface to quickly transfer cell type labels from different cell typist models to your data of interest, which will also facilitate cell type annotation for users without encoding experience. So please try it out here at this website. And of course, a good model should be very efficient. And in cell typist, this is achieved by two means. First, we introduce mini batch training, where 1,000 random cells were used as a single batch, and 100 such batches were trained in each epoch. This will avoid loading the entire dataset during training, and after 10 epochs, allows the classifiers to see the numbers of cells with 6 orders of magnitude. In parallel to the mini batch approach, the cell typist use the stochastic graded descent learning to solve the loss functions to allow online training. The combination of these two approaches yield promising results in the performance of cell typist. As you can see in the plot, the cross validation performance quickly converge within 10 epochs even we do not solve the loss function for the entire dataset. And finally, a good model is supposed to be accurate. In cell typist, we implement a whole optimization protocol to optimize the performance of cell typist. I will not detail each element here, but would like to highlight 3 critical modules. First, the core classifiers, regardless of logistical regulation or SGD-based logistical regulation, will undergo RL2 regularization to result in non-sparse solutions and to avoid overfitting. And second, the query dataset will borrow the scaling parameters from the reference dataset, meaning that they will have comparable expression scales, and the L2 penalties will not be skewed by their expression scales. And finally, utilizing the cell-cell similarities, cell typist applied an over-clustering followed by majority voting approach to harmonize cell type structures within local clusters. And now let's turn to the real-world applications of cell typist. This example came from our lab, where my colleague Cecilia and the collaborators Laura and Daniel extracted 330,000 immune cells from 12 tissues of 12 donors. And based on this, we asked, can cell typist resolve this degree of heterogeneity for these immune cells? And using cell typist, we can annotate this dataset into detailed immune subpopulations, including T, B, and myeloid cell compartment, and their detailed effector and effector phenotypes. So, based on this, we anticipate that the application of cell typist will be most useful when a large degree of heterogeneity is found in your data of interest. A quick summary of this part. We developed cell typist to annotate cell types for the single cell data. And cell typist is not only a tool for automated, precise, and rapid cell annotation, but also a framework for data-driven cell annotation system. And initially as an immune cell database, now cell typist and its models have been expanded to different tissues and developmental stages, covering more than 800 cell types in the human body. And we also welcome you to try our cell typist website and the Python package to see how your data can be annotated into what cell types. And now we have used cell typist to annotate cell types. The next question is, how can we unify cell types from different datasets? This is a very critical need because, as we all know, different labs have their own ways to define cell types, and we need to find a way to combine their different cell type definitions. And to this end, we developed cell hint to harmonize and integrate independently annotated cell types across different datasets and laboratories. Specifically, cell hint has 2 main functionalities. The first is harmonization, meaning that we try to establish links between cell types of different datasets. And the second functionality is supervised integration, meaning that we try to place the same cell types obtained from the harmonization step into a unified representation. To achieve these 2 functionalities, we need to overcome 3 technical challenges. First, how to robustly measure cell and cell-cell distances with minimal influence from the batch effect. Second, how to align cell types of multiple datasets and to summarize and visualize this information. And third, how to incorporate cell-cell relationships into the single-cell data integration process. For the first challenge, The cell type is cell hint built on the predictive clustering chain algorithm. For those of you who are not familiar with this algorithm, it is a multi-target regulation chain algorithm where cells are iteratively split into descendants until they form some homogeneous groups. And the two key elements here are the split and the cell groups. For the split, it is executed by a specific gene in the feature space. Because here only a specific cutoff is used, meaning that the algorithm is robust to expression scales because only ranks are considered here. And more importantly, for each node or leaf, the similarity of cell types are not measured by the expression profiles, but by their similarities with all cell types within the same batch, creating a multi-target regression scenario. This means that when a predictive cluster tree is built for each reference data, a query cell will be predicted into the same space. This has a direct consequence that cell types from different datasets will be placed into a common graph to more accurately measure their cell type to cell type distances. And the next is the second challenge, how to align cell types of multiple datasets. This is even more difficult because, for example, when you have 4 datasets, you will have 6 pairwise comparisons, totaling to 12 directional label transfers. How to summarize this variety of information and visualize them in a 2D plot is not resolved in this field. And the approach we solve this problem is iterative cell type harmonization, consisting of 2 modules, pairwise alignment and visualization. First, because we already have robust cell-to-cell distance measures from the previous step, we can align cell types of 2 starting datasets, as shown here. This will result in several situations, including 1-to-1 alignment, 1-to-multiple alignment, multiple-to-1 alignment, and unique cell type in a single dataset, accompanied by visualization of this plot symbolically. And the next is the key step of merging cell types. Specifically, we can put together cells of 1-to-1 alignment and split cells in the first dataset into different subtypes based on the 1-to-multiple alignment, and also repeat this process for multiple-to-1 alignment. And after this pairwise alignment, we obtained 6 new cell types from a single merged dataset. And now comes the third dataset. And if you observe again, we have 2 datasets again. We can again do pairwise alignment. And after pairwise alignment, we can also extend this graph to add more cell types. By gradually adding more datasets and more cell types, we can integrate cells from different datasets into the previous cell type structures, accompanied by extension of this graph, which we name as the harmonization graph. And finally, the challenge is quite obvious. That is how to utilize the above 2 information and incorporate such information to accelerate single cell data integration. And the strategy cell hint used is restricted neighborhood search, comprising 3 steps. First, for each cell, we find its original cell type annotation, and then we locate the cell type branch according to the harmonization graph. And finally, we search neighbors for these cell types based on the harmonization graph. For example, this blue cell will only search neighbors in the second and the third datasets because the harmonization graph indicates that the fourth dataset does not have such cell annotation. So by this approach, the search space is geometrically narrowed down, and the same cell types will tend to stay together. So now we have solved these three challenges. So how can we benefit from these fruits? I will detail an example to show the practical applicability of cell hint. This example is focused on the adult hippocampal neurogenesis, where newborn neurons will be produced in the dentate gyrus of the hippocampus and gradually mature into granule cells. This is an important process in most mammalian species with potential implications for neuron circuit plasticity, stress response, and pattern separation. And the single cell technique is an ideal technique to detect such phenomena because a great number of cells will be measured in a single experiment, facilitating detection of real cell population. And until now, based on single cell transcriptomics, a complete neuron genesis trajectory has been detected in adult hippocampus of mouse, pig, and monkey, starting from very early immature neurons to the mature granule cells. However, based on single cell transcriptomics, no such trajectory has been found in humans. Although some studies claim to find such populations, they either detect only one such cell, which was not reliable. All these cells can be only detected by machine learning-based cell scoring and were scattered across the granule cell cluster and did not form a robust cluster. This means that we need a tool which is more powerful and interpretable to enrich such signals, and the cell hint presents such opportunity. And to this end, we collect 6 adult hippocampus human datasets, which after quality control resulting in 700,000 cells used for downstream analysis. And next, we applied the 2 modules in cell hint, including the cell type harmonization and the supervised integration, followed by manual inspection and revision to refine the cell type results. This simple approach works quite well because, as you can see, the batch effects from different donors were significantly mitigated. And more importantly, using cell hint, we can detect 51 cell types and subtypes in the adult human hippocampus, including excitatory neurons, inhibitory neurons, glial cells, and their detailed subtypes. And we indeed detect the immature neuron and the neuron blast populations in this dataset, as shown here. This population uniquely expressed markers relating to immature neurons or neuron blasts, such as D6, SOX11, SOX4, BHLE22, as well as other markers, which were together enriched in neurogenesis-related signaling pathways. To further confirm this discovery, we used the cell hint to harmonize 5 additional datasets from 3 mammalian species. By cross-species cell type harmonization, you can see that there is a good correspondence between the of the neuron blast populations between adult human, monkey, and pig, which were further subdivided into 3 subtypes in the mouse. So this result further showcases the utility of cell hint in cross-species cell type harmonization. A quick summary for this part. Cell hint is a tool for automated cell type harmonization and integration, particularly designed for assemble independently annotated cell types across datasets. Until now, we have used cell hint to establish 12 organ atlas, and we plan to extrapolate cell hint and its pipeline to different scenarios. And that's all about my presentation. In short, we developed cell types and cell hint to annotate and unify cell types. And I would like to elevate this to a higher level, not only about making full use of the single cell data, but also about the entire community where we bring together everyone's cell type knowledge and to make sure every member has the opportunity to benefit from the collective human progress. And there are a lot of people I need to thank. And among them, I would like to particularly thank Sarah, who guided all this work, to Cecilia, who worked together on cell typist, and to Martin, who provided the great technical support, and to donors and their families, and to the funding bodies. And finally, thank you for your attention.
Thank you very much for a really thought-provoking talk. I think there's a lot of elements in there that a lot of people will be interested in. Now, at this stage of proceedings, we're going to open up for questions for either of the speakers. Please put your questions into the Q&A box rather than the chat box, and please let us know which speaker you're directing the question to. I believe, Ben, we have a question for our first speaker, Song. Is that right? Let me see.
Yeah, so this question comes from Trevor Freeman, and it asks, how does your method's performance compare to deep learning-based cell type assignment methods, such as those available through SCVI?
Okay. So I didn't use SCVI myself, and to my knowledge, there's not a cell type assignment method. It's like a deep learning based imputation. Can you hear me? Right.
Yes.
So I don't know the method, and I can take a look. But to compare to their method, the one advantage is our method is very simple. It's like a co-enrichment for cell type marker genes. That's it. And I'm not sure that's the advantage or disadvantage, but we just need a list of markers. The key thing is that when you call cell type with markers, so we want to advocate that you want to do this statistical test across all the markers, all the clusters, to ensure that the things you called is actually correct.
Cool. We also have, while we wait for other questions to trickle in, we have a couple of broad questions, I think, for both of the speakers. And maybe you could sort of tackle this. The first one is, I guess, pretty applicable. How broadly do you think your research approach could be applied to other organisms? And this is sort of like more broadly outside of the list that you already explored.
So can I talk first? Yeah. So the answer to that is I don't know yet, because we haven't tried it in other species. And to my knowledge, if you work with human mouse, for example, the one-to-one ortholog genes are pretty well known in that biomedical research community, so it's not a major problem. But there are other people working on different other species, like insects, like Drosophila, right? Mosquitoes. Maybe those are the communities where this OMG approach, like calling these marker groups instead of one-to-one ortholog, could work. And one of the reasons I think the also groups is working better than one-to-one ortholog is because plants is well known to have this gene duplications, and then whole genome duplication, many reasons that plant duplicate their genes, and then they have multiple parallel copies, and then their expression diverges, but their sequence are still quite conserved. So that's why we want to do this also groups. So maybe in other species there are similar scenarios happening, and this is useful. But to my knowledge, again, between human and mouse, is probably not necessary to do this. But we don't know. We can give it a try.
Yeah. So for like cell type transfers among different species, it's highly reliant on the ortholog genes, in particular the one-to-one orthologs. For one-to-one ortholog, it's easy. Like the query dataset just use the weights associated with that ortholog genes. But if it's one to multiple alignment, then you need to choose some weights. For example, one approach was to choose the ortholog with the highest weights, and discard all the other ortholog genes, only focus on the ortholog with the highest weights. And the other approach is to average all ortholog genes to make them some meta gene, maybe this name to use as one-to-one orthologs. But anyway, it at least for cell typist and cell hint, it relies on the ortholog genes, and we mostly transfer cell type labels between humans and mouse. It works quite well, at least for immune cells, but haven't tried more distant species. Still pending verification.
And there is another question for you, John. For your iterative cell type harmonization, how robust is it for the input references or starting data that you use to add cells to? I'm just looking to see. Chris, was that your question? Did I?
Yeah. I mean, so I think you asked it. I mean, the main question I have is, you're starting with a particular data set that you add cells to. So if you change that data set, how robust is your output?
Overall, it's robust because it's based on the fact that most published studies have good annotations. That's a very basic assumption. So even you start with two data sets, their cell types will aligned not bad. And then when you add more data sets, usually it will converge to a very stable picture. But we do see some scenarios where starting data sets are low quality, but overall it's good, I think.
So you normally would start with a reference, or do you start with a single data set?
Usually start with more comprehensive with data sets with more comprehensive cell types, because it will make a long time.
And perhaps I could follow up on that with a question. Your cell typist approach appears to start with quite a large single cell data set coming from those big projects. So how do you think it would work if we had much less starting data, as we're going to see in a lot of these other species? Do you have thoughts on that?
Yeah, so big data is always good because you have good coverage of cell types. But for small data sets, some cell types may not be covered. So if your cell type is to predict the query data, some cells will output low probability, meaning that we cannot find corresponding cell type in the reference. It's probably a novel cell type in the query data set with low probability. So there are some indications to show such phenomena. Yeah.
Okay.
Another sort of general question for both speakers is, if you had to start all over again, either go back in time or start on a different species or a different system, what do you wish you had? What resources or information would you want in the ideal case?
Yeah, so to me, we had a lot of trouble sort of make the exact UMAP cluster people published, because some of the detailed parameters they didn't provide in the publication, which is understandable, and because you don't know whether that is necessary. So if you go back, I would say if sort of regeneratable code for your cluster plot would be really helpful. So we end up using the Chinese group. They curated all of this and put in their database. Format is really nicely. We start with by doing this ourselves. It's a very challenging process. So if there's sort of a GitHub code where you have one click regenerate your plot, that would be really helpful. I think there's some services like that now exist, but I don't know exactly what it is.
I think this raises a very good point about the reproducibility as the community starts collecting this data. Exactly what do we need to be measuring and recording so that we can make this reproducible, right? Yeah. There's another question in the Q&A, and this is for both speakers, and it's from Trevor Freeman. Do either of you have any comments on how your methods perform when transferring labels between libraries prepped from different single cell technologies or platforms?
Yeah, so for cell types and cell hint, it's overall robust to different techniques because only like as I mentioned in the slide, only not so many features are selected. So even you have different techniques, these features are like some like marker genes, and they still point to the identity of the cell types. And also, although like cross technique label transfer will result in low probabilities overall, but they have some trend to tell you that even this match is low probability, it's still the best match. So it can tell you, at least tell you some information across platforms. Yeah.
In our case, the second half, we show this pattern infection that was single nuclei RNA-seq and it works between single nuclei and single cell. We don't know if there's anything other than there's a parse Bell-Science, anything in plants published, I don't know. And there's a PIP-seq or pipe-seq. I also don't know whether there's anything published from those newer platforms. So that we don't know. But nuclei and RNA-seq seems to be okay in our. Which I mean, when I say RNA-seq, I mean the protoplast-based single cell and the single nuclei seem to be okay in our case.
Another question from the Q&A for both speakers. What future directions are you interested in? For example, advancing your models or improving data set handling?
Yeah, maybe I will start first. So I have some feature directions for cell types, but at least now it sounds too ambitious for me because I want to understand what is batch effect. Because people usually say that two datasets are from two labs or from two different processing dates or protocols, then they have batch effect. But what is batch effect? Mathematically, it should be something like meaningful rather than some ambiguous concepts. So I think now it's like the era of big data, and people try to model a large number of data sets, and the noise is very big. And with I try to understand what the noise is, and then it will make this direction more clear for me. But like I do not have a detailed plan, but that's my thought on this. Yeah.
So the question is about future directions. So I think on my side, I really hope that more plant scientists in other domains can pick this up. But then I don't know how to bridge this. For example, breeding people when they do GWAS, why do you need to know where this gene is expressed in which cell? How do you put that into your breeding pipeline to understand gene functions? And so that is one thing, because I can see many people are interested in doing single cell sequencing in many different plant species, but there are also people that sort of don't know how to use this information. So that's my interest. My student interest is to build this AI large language model based off things to assign the also groups. So instead of, because we only have 15 species, every time you run this also finder, it gives you the different cluster if you put in 2 more species, right? And then she want to solve this by language model, just give a language model is tell you which cluster it should go to. So those are the 2 things.
So I wonder if I could follow up on that, maybe for both speakers, but is there plans or is it possible to use the tools that you have to look at genetic effects on prediction of cell type? So that could be, something that would apply to breeding, but also just to the methods. How much does genetic variation potentially affect these cell predictions, or are we expecting it to be robust to genetic variation?
So that is a very good question, and the limitation is the cost of generating data across different, variety of plants. I don't know there's any. Maybe there's one from Arabidopsis a couple of years ago, but I don't know there's anything out there that we can use because it's still pretty costly, right? And so yeah. So that's my take. And maybe you can do some imputation, maybe pick a few one that with very distinct genetic makeup and then sequence those and see maybe someone already done that. Right? Yeah. But again, it's the cost and what to sample is the issue. But that would be very interesting to see.
Well, what might be one thing that we talk about next week in terms of how do we move toward genome to phenome. But one of the things that is interesting to some groups is using the single cell data to then deconvolute bulk data.
Okay.
And my concern is that if we develop a deconvolution tool based on limited genetic variation samples, then is that going to seriously affect the ability to predict cell type in other genetically diverse individuals in that species or across species?
I think it also depends on what resolution you would like to go. Yeah, if you only want like cost level, then deconvolution will always tell you like blood signals, blood cell types. It always will be accurate.
Thanks.
Maybe a last sort of broad question is, what do you think the current big picture challenges are, in your approach, or and are there ways that the community can help? Are there ways to sort of crowdsource around some of the big challenges that you might be facing?
So I think for cell type prediction, one of the biggest challenges is about the homogeneous cell clusters, which is like for manifold, and it's different people have different ways to split the cloud into subcloud something, and especially for computational analysis, because even you do not notice, the result of computational analysis will always biased to what you want. So it's very risky, especially for analyzing these populations. So I think here we need more knowledge. Like there are some well-established criteria from experts who can say that this, like the differential process or these cell groups should be like this, and we should find a way to incorporate the knowledge into the model and to have some very like near perfect prediction. And I think it's quite big challenge in cell type of prediction. Yeah.
I agree with the part of the comment is about homogeneous clusters. Different people split them differently. And how do you get the cell type out of those clusters, right? And then that goes back to how do we document this process and how do you make decision. And another thing to me is that although many of us can do the single cell analysis with smaller scale data on your laptop, is not still super accessible to people. If you want to look at all these 1 million cells, that will be hard computationally. You cannot even load the data. The UMAP is very slow. So that's something I think the human community may have much better cloud infrastructure solve this partially, and we don't have that for the plant. to my knowledge, that can do it quicker. And so one solution I could just put out there is like, if you do some sort of autoencoder and encode everything, become much smaller, and you can generate the data on your computer on the fly instead of every time you load the cloud database. That could be something interesting. So just like everybody can browse this data and look at the data is the limitation I see.
Following up on what you said, John, then is it that we need reference sets to or reference sets with workflows to say this is the standard way we will do this? Of course, it won't work for everything, but if we move from this idea of a gold reference standard, perhaps to a benchmark set, and then with that benchmarking, you're able to adjust and see what the changes are, what the differences are. Is that a better way of thinking about how to do this, how to answer those questions?
Yeah, I think so. Also, there are other different situations like the healthy versus diseased, like cell types, and like there are different scenarios, and you need to have build like very specialized reference atlas for a specific question, and the complicates the question. And yeah, I think so.
Yeah, I think that's along that line of thought. There's a Busco score for whole genome assembly. If you do microbial there's something called CheckM.
Yeah.
We probably need some sort of thing like that, right? So Busco score for single cell sequencing, and then with some benchmarks, and if you miss some of this, then you probably need to be careful with your interpretation of the data. But I don't know whether that exists or not for the human. Usually, that if anything like that is already there, it's probably already done by human researchers, right?
Yeah.
And I don't know enough about that.
A Busco score for a reference data set.
Yeah,
maybe.
For the single cell.
Exactly.
Yeah.
And I think sort of getting to what that score would be, or quality score, I guess we could call it in general, I think that comes back to what are we expecting, what can we reproduce? And I think that will come back to, we won't all agree on the same analysis because it won't work for everyone. But if we have a benchmark set to start with, you can start seeing how yours compares to something else, even if we can all disagree that that's the best benchmark. So, yeah.
And we need the benchmark has to list all available cell types.
Well,
Fiona, that's going to be tough.
It needs to be, if you can make it a certain data set, and you know that, yeah, you'll need multiple benchmarks. But I mean, I think you got to start somewhere, right? And perhaps this is where we open up for a broader community discussion on what is it that you are looking for in your data sets. And one of the questions we had for everyone in the audience, and I guess we're letting Song and John off the hook a little bit here, but the question we had is, how confident are you that you will be able to assign cell identities for the organisms or the data types that you're working for?
So usually I will cross-check with different data sets, because so to get a sense of how different data sets assign these cells, and then to combine their results. And this also involves a manual curation process, which is painful but worthwhile. Yeah.
Yeah, I'm just thinking. So, but if you look at the big heat map we produce out of 260-ish cell clusters, actually a lot of them are correct. Even there's a lot of flaws in how you actually annotate. And one of the things we noticed that if people pick a cluster, they always go with the biological function of the marker gene. So sometimes if the marker, like the scarecrow, for example, is endodermis marker, been confirmed in many species, and then you really just need that one gene to decide. Right, so that's where I think, yeah, what I was trying to say is that, sort of, from my perspective, if you do this meta-analysis, you generate bigger clusters, and then across many species, and then they give you increased confidence that the cell type assigned is actually correct. And then that can be helped to see maybe some cell type goes to the wrong cluster, but then maybe they contain some unique biology there that they just fall into endodermis, but they're different. So I think, yeah, I don't even know what was the question, but yeah.
And Trevor, I see you've added a comment into the chat. I'm not sure, was this in reference to the discussion on the standards and benchmarking, or was this something separate? Are you able to, I don't know if you're able to unmute. The comment was, is that something that you could produce with the single cell LLMs that are being produced? Could you use it to produce some sense of how unlikely it is to see cells that look like the ones you produced, for example?
I think it was in reference to that Busco thing.
Yeah, exactly. That's what I think. I'm not sure if Trevor's able to unmute.
So that one thing I know in the human literature research is they do this thing called foundation models, where they train this big model on millions of cells and then start to use that to basically generate the embedding of your cell when it comes in, and then sort of works like a ChatGPT type of thing, and can give you some sort of score used for some downstream task. But for plant science, the challenge will be we don't have a million cells from our Arabidopsis. If we put everything together, it's about 2.5 million cells. If one of Joe Ecker's data got published, we get a little bit jump on that. But then if we include the ataxic data, then we get a little bit jump. But we don't have as much as the things that are used for train the foundation models in human. And also the cross species, the foundation model mostly trained on human mouse, rely on one-to-one orthologs. But for plants, we don't have that many cells to start with.
I think that's going to be true of everything outside of, the human mouse, maybe Arabidopsis, right? So it's just going to be a recurring problem that we're just going to have to do what we usually do and figure out how to work around it. So, yeah.
Maybe a penultimate or last question for Song. Do you observe a point of diminishing returns after incorporating a certain number of species or tissues in a plant?
Oh, very interesting. So I don't know the answer to that yet, but I think the what I'm looking for. So there was a question about what we look for in the future. Is that actually more diverse tissue types, like the things we have not seen in the current literature, like some specialized flower tissues, for example, and maybe lateral root initiation process. So into those more biological interesting process, not just meristematic, really young 5-day-old Arabidopsis root tips or many. Well, those are interesting. Take that back. Those are very important first steps, right? But then now we move into the cases where it's become more challenging to get the single cell done. For example, in wooden tissues, how do you get nuclei out? There are papers from Matthias group showed some early effort on doing that. So I'm looking for diversity of cell types, but I don't know where's the point of. So another way to address this is, how do we know, the cell space for all the plant species, right? So we have maybe 15 to 20 cell types in roots in Arabidopsis, but then if you think about other plant species, what are other new cell types are there? Can we strategically decide which species that we need to go and sequence it, and then just build this space, and then other things can just be interpolated computationally, or you don't have to sequence that deep for other species. Just you get this cell space for all plants. That would be really cool.
And perhaps to follow up on that train of thought, a question I have for both speakers is, knowing what you know now and going back and thinking if you wanted to redesign or develop these tools again, what is it that you would be looking for in terms of resources or data sets or those kinds of things? What do you think that was missing that would have really helped you when you were in the development stage? And you can't just say more data.
So I think one thing I have missed for the tool is that sometimes I focus too much on the algorithm details, but like ignoring the quality of the data. I usually like relying on finding high quality data, high quality published data, but sometimes you need to have like better quality because like good quality data will produce good results, and if garbage in them, garbage out, and especially for cell type transfer, actually the most critical point for cell type transfer is the quality of the reference data. If you have a good reference data and whatever algorithm you use, logistical regression, random forest, or some deep learning ones, you will always have very nice prediction result. So I think curating data and of course more data is very important. And I should like focus on this in the future.
Yeah. Okay.
Yeah,
I'm gonna ask a. Go ahead. No, go ahead, Sung.
No, I don't have an answer to your question, so go ahead.
Well, I just was thinking about the question about how do we have enough, how do we know when we have all the cell types? And I guess I was just thinking, where can we start with that? I was wondering if embryonic plants and embryonic animals or fetal, at some point, thinking about a developmental stage approach where at least you would get the starting early progenitor cells of all of the cells in the organism. I mean, should we focus on very early developmental stages for that?
Yeah, and I sort of wanted to follow up on that, not to get too metaphysical here, but I think in the plant cell atlas community, we're also having a discussion about what is a cell type, how do you define it, and like what, yeah, exactly, what is it? I mean, like at least in the plant community, they've almost all been defined by like a tissue and anatomical set of features and maybe some physiology as well. But in yeah, I don't know what the case is, and I guess it's all physiology in animals and
same problems. Yeah.
Yeah. But yeah, like how do you delineate like what is its own, what deserves its own name, I guess, in the cell type lexicon. And I think if we can answer that question, we can maybe start to answer how many, how do we know that we've got them all, I guess.
So it's 11:30. I don't know.
Okay.
Maybe you want to end on that philosophical note.
Yeah.
And the
yeah,
unanswerable question, right? Yeah.
Yeah, and I agree with the sentiment from Trevor. That do you think we get too hung up on trying to identify cell types? And yeah, probably. But I think it's an important question to keep asking ourselves, periodically as we explore these new technologies. I don't know if do you want to close us out or?
Yeah, I'd like to start by thanking both of our speakers, not only for great demonstrations of the tools and resources they've been developing, but also for agreeing to do this in this format, which I know is challenging, and for being open to really sort of thinking about these questions and helping us with this process. I'd like to thank all who attended, and hopefully we'll see many of you again for our final session, which is same time next week, right, Chris? Let me make sure I get that right. Okay. And with that, thank you, everyone.
Thank you.
Thanks.
Thanks all.
Thank you.